Hi, I'm Helen. I'm Dr. Helen Ragel, but you could just call me Helen. I don't want to torture you with pronouncing my last name. And I'm here to talk to you about biocompatibility, the most exciting hot topic out there, in my opinion. So when I talk about biocompatibility, oftentimes I get this, kind of puzzle faces in some of the audience. It's like, what is it? And we will talk about that. Fear not. We'll go over what it is. But there's also a couple of other things that I wanted to focus on in my talk. I wanted to go over a little bit how to perform a biological evaluation, so you get an idea of what goes into it. And I wanted to point out some very important pieces of information from these two master documents, the 10993-1 and the FDA guidance document on the use of the 10993-1. And then, the main point of my talk will be the third point, is when do we need to start thinking about biocompatibility? And we'll talk about that too. So you will have an idea. Hopefully later you'll be like, thank you, Helen, for reminding me. And then, why is it moving? I'm not even touching it. Okay, and then we'll talk about changes, right? The headache of every manufacturer out there. Changes to the device is already out there, years and years. And then you hear there's a supply chain issue, or your marketing people are like, ooh, let's make that pink, right? And you're like, oh, okay. So we'll talk about that a little bit too. Now, first things first, what is biocompatibility and why do we care, right? Biocompatibility in an essence is do no harm, right? Like the oath that the medical doctors take, this do no harm to the patient that the medical device is going to be used on, right? And so I always say, I truly believe when a group of people, they sit down together and they try to design a new medical device, and it's supposed to cure this or make, the quality of the life of the patient better, right? They have good intentions. It's a good intent to do something like that. And they want the outcome to be good too. However, sometimes we know that good intentions don't necessarily always translate to great outcome. Happened to me many of times in my childhood where I had very good intentions, but then it somehow didn't turn out the way that I really wanted to. Many times. So I should, cut my kids some slack, right? So what we want to do is we want to make sure that before the device goes out there, we check its function, it's going to do everything that we're saying it will be doing, and then it will also be safe, right? Safe for the patient. Now, another point that becomes a problem for biocompatibility is what happens if something goes wrong with my device, right? For example, we've had a scenario one time where the surgeon came back, said, I used this tool during my surgery, and then after the surgery, a part of it is missing, okay? And then he's like, okay, it could have ended up in the patient, right? So now we need to be like, okay, what part was it? Is it better to leave it there for the patient, or do we need to butterfly the patient up again and gauge it out, because that would be, worse to leave it there. So that's something you have to think about, right? And then what happens if there are changes, right? Like I mentioned, if you have, let's say you use Mucil, right? Everybody loves Mucil. And now they're saying, oh, sorry, we have a back order. We can't give you the one that you want. What do we do, right? I mean, I think everybody knows that situation. And after you go and scream in the pillow, you have to come up with an answer or solution, right? So we'll talk about these things and how they impact biocompatibility. Now, I always say that we need to think about it. I'm a scientist, right? And I need to be able to make sure that if I do testing, I need to know why am I doing testing, right? I was that annoying kid in class, always asked why. Well, now, serves me well. I always ask the why, okay? And when I was expecting, my husband said, this is the greatest book you need to read, right? Anybody read it? Anyway, the gist of it is that you need to look at your child and you need to find out what is important for them and what is important for you. And the stuff that doesn't matter, you just gotta let it go, okay? So I say we need to be doing biocompatibility evaluations also with love and logic, right? There's definitely some risks that are associated with some certain devices that need to be, looked at, addressed, maybe with data, maybe with more testing, but we need to know and focus on those and not the stuff that doesn't matter, right? Now, as I said, let's say, okay, you come to me and say, okay, Helen, I don't know why it's doing that. It's kind of flickering, and I'm not even pushing the button, okay? Okay. All right. I got a fancier ones. Okay, so let's say you come to me, Helen. I had this magical medical device, and it's supposed to make the patient live long and prosper. Okay. And then they say, okay, Helen, what do I do? And I say, okay, that's cool. Let's sit down, let's discuss what is it for, how it's going to be used, right? Because the first question I need to ask, how does it contact the patient or the user, right? Because if your live long and prosper device is going to be just handheld, right? Like this one, right? It carries much less risk than if you say, no, Helen, it's not carried, around, because you can lose it. You don't want to lose your prosperity. So you have to implant it right next to the heart. Okay? So you can see how if that's the case, there's so many more risks that we need to consider when we do the biocompatibility evaluations, right? So another thing is, are there any special considerations, right? For example, if you want it to be implanted next to the heart and it's a bioresorbable material, right? Because those are hot things right now. And you want it to be bioresorbable so that it will be becoming one of you in your bloodstream, right? That will give you even better force. Then there's other things that you need to consider, right? Like how does it, where does it end up? Is it going to be toxic when it breaks down? All of those special considerations need to be thought about, right? Another thing, if you say, okay, well, I can see that it's 3D printed. Okay, then the 3D printing, another very cool technology right now, up and coming. However, we know that when you do 3D printing, you have lots of potential particulates from that process. So now we need to make sure that there is not these particulates on the final finished device, because they could also cause harm to the patient, right? They're not part of the device, but they could be implanted into the body with the device and cause harm, right? Another thing is the structure, right? The structure of the material on the top. We all know the fiasco of those breast implants, right? It basically boiled down to that structural thing that caused, chronic inflammation, and that led to, cancer. So really, these are all the things that we need to think about when we think about biocompatibility. Now, when we are, doing biological evaluations, the question is, what risks are present based on the type and duration of contact? Again, like versus handheld versus, implanted. And then the bigger question now is, how are you going to prove the safety, right? Yeah. And before it was like, oh, it's an implant. So, here, these are the endpoints, test for it. You're good, go, right? This is not how it's done now anymore, right? Because sometimes when we do just that, we just follow the letter to the T and do exactly what is asked for, we might miss some important aspects, right? Like, for example, I always give this example, like, let's say I have a delivery device that injects something in the eye, okay? It's a very small needle, right? And contacts for like 2 seconds, okay? So, and it's stainless steel, okay? Really, really low risk, right? Because we know a lot about stainless steel. And if it's such a short contact time, stainless steel probably passivated, very common method for treating metals. And then, but if we inject something in the eye, I would be worried about particulates, right? Because I don't want anybody to inject some particulates into my eye or the patient's eye or your eyes, right? So that's something that is currently even not mentioned in the 10993, right? But it's definitely a risk that we should be looking at. Another point is that when we do the biological evaluations, right? Love and logic. We need to look at what available information do we already have, right? Let's say we have a catheter, right? And it's we're developing a next-gen catheter, right? And we have all of this previous testing and available information on the first gen. Do we really need to test the second gen again? Let's say if you're only changing the proportions of some of the sections within the catheter, I would say, spoiler alert, no. But you have to do that mental exercise to make sure that everybody understands, yes, this is why. We are not testing, dog thrombo again, because it is not value added. We have available information to cover that risk, right? So that is really what the industry is moving towards, having that mental exercise, right? In the form of a biological evaluation plan, right? Now, and then, when you do review all of the available information, and you look at all the data you have based on the risks that your device specifically carries, your child, and know your child and know the risks, and how are you going to address it? And if there is gaps, obviously, there might be need for more testing, right? But we shouldn't jump to the testing part right away. Okay, that's the main thing. Okay, now, this is the famous Table A1, right? And you can see lots and lots of different biological endpoints that are, listed there. And then it even says there could be others, right? Now, the difference is here, it says E, right? I love the E because it stands for evaluate, right? It's not like X, you got to just do the test and you're done. You have to make sure that you evaluate it, but not necessarily test for it, right? So again, a written document that covers the evaluation process would be where you do this evaluation. Please sit down. There's tons of seats here. Okay, so another great thing, a beautiful morning in October, I think it was October 8th, 2023, FDA updated their guidance document. Do you remember that day? It was a beautiful day. What they really did was they added an attachment G to their guidance document. And that attachment G says that based on historical data that we reviewed, we believe that certain materials are safe for intact skin contact, right? They probably read like 50 billion of my risk assessments that said, please do not test this thing for irritation and sensitization. And they were like, stop, Helen. And then, they put the attachment G in it. Anyway, so basically what it says is that we know from previous experience on medical devices for intact skin, ABS, for example, polycarbonate, all of these widely used materials. I mean, we also use like nylon and like cotton and all of these clothing things, right? That we know carry low risk, especially when it's just in contact with skin, because skin has, its natural barrier properties that make it, less likely to be systemically exposed. So, it totally makes sense. So now they say this is the least burdensome approach. You do not need to provide testing reports when you can prove that your device is intact skin only and has been manufactured from one of those materials that are listed in the table or in that attachment G, and that it supports the principles of the 3R to replace, reduce, and refine animal testing, right? Okay, so I thought that was a great step forward. It was a beautiful day. Okay, so for biological evaluations, the main thing you need to do is you understand that there has to be a plan, right? I have had many customers that come in and they say, okay, we have this device and we need these tests. And I'm like, okay, well, why? Right? And then they're like, well, it's because we've done it always like this and we're just going to do that same thing over and over. And I was like, yeah, did you know that the 10993-1 says that you have to have a plan before you go into testing? So I do say, we always have this saying, like, if you didn't document it, it didn't happen, right? Quality management, yeah, exactly. If you didn't document that you had a plan and you have a path forward for evaluating your device, then it didn't happen. That means that the reviewer or auditor or whoever, they're going to be like, I don't know if you went through this mental exercise. I need proof, right? So really, the plan would be something that contains information about your child, your device, right? And it talks really about how it is used so that everybody understands. Sometimes I read these documents and they're very technical, and I say, please try to put it in the words where everybody can understand, because the FDA reviewers, they're smart people, but then, if they're moving from like some blood device to something completely different, they might not know all the specific language. So you need to make sure that the reader follows what you're saying, okay, and can understand what is it that you're wanting to have them read and understand. It really is about the story you tell about your device, right? And then you have to demonstrate that you know exactly what goes into your device, right? That's the first column of the 10993-1, Table A1, physical and chemical information. I read the guidance in a way that it says you have to know exactly what goes in there and how it's made, so that if you have any changes, that will trigger your QMS to be like, hey, whoa, we need to look at this, right? And that is the point of that. And then, obviously you want to show the available information that covers like the rationale. Are you going to use available information to justify for not doing certain tests? How does that cover those risks? And then in the end, it really states, okay, based on everything we know, these are the gaps, and this is the approach for testing that we're going to do if testing path is chosen. I also say that like the medical devices that I keep on seeing, 5 years ago, they were so much simpler. But now, people are coming up with these creative solutions, all of these the new materials, but with those, it becomes more complex. Even if you have to do testing, that testing can be very complex, right? And even the FDA says that if you do have a very complex device, make sure you discuss it with us before you start doing these extensive, expensive tests, so that we don't have to do it again, right? That's not in anybody's, yes, Nelson Labs, for example, we do testing, right? And, okay, if it happens to be that we have to test it again, so be it. But we want to be able to provide you the one test report, and, this is the thing, and you provide that, and FDA says, okay, that's what we really want, right? Okay. Now, these are the different tests that 10993 series, the bazillion series have, right? There's biological tests, there's analytical chemistry testing. All of these are available at your hand, at your service. But what I say, you have to look at these tests as tools in your toolbox, okay? You need to know what the test is testing for, how, like a little bit how the test is performed, so you can understand, is that the right test for you, right? Is it covering the risks that you see, and so that you can close the gap, right, for patient safety? So talking about tools, the one person who taught me the importance of tools is my husband. He's obsessed with tools. And I kid you not, every single time we go to Home Depot, we cannot walk away with buying yet another tool, okay? So, obviously we had to move to a bigger place to accommodate all these tools. I'm not saying you have to be so cray-cray about tools, but you should know what the tool can do for you, right? Because let's say you want to hang this beautiful picture that you got from the store on your wall, right? Now, do you need a power tool to do that? Probably not, right? A simple hammer and nail would do. Or maybe in your circumstances, a command strip will be a better choice, right? So know what you need to have that test answer before you go do that test. Does that make sense? Right? Love and logic. Okay? Now, when do we need to start thinking about biocomp? When you think about all of the guidance that we have from the FDA guidance document and 10993-1, don't read those. You don't need to read them. Basically, what it says is final finished device, right? I mean, how many times have I had this conversation with the FDA? We only look at final finished device. And it's like, final finished device, right? I mean, sometimes I go into bed in the night and I hear them say, final finished device. Yeah, okay. So biocompatibility, it has to be an evaluation at the very end, right? But I want to make sure that this point is really clear. We cannot think about biocompatibility as an afterthought, right? It's because what happens if you go all this trouble, you have this beautiful device and everything is great. Now you're like, okay, Helen, we just need the testing and then we'll be done, right? And then we can submit. What if something goes wrong, right? All of this effort up front would be useless, right? So really, when you think about final finished device, that's like you have your materials, you have your processing steps, and then you get to the final finished device, right? There. Well, no, this one doesn't do it. Anyway, there. Okay. But again, please don't make it as an afterthought. Even the document says, if you like go deeper into the document of the 10993-1, it says material selection should be a key design input, right? That's why my title of the talk was Key Design Input, Biocompatibility, right? So what we need to do is really think about it up front. Okay, so I wanted to give you this kind of like this story that happened about 5 years ago. And this is not something that happened to, I don't know, my childhood's friend's uncle's neighbor's kid. Okay, it's not that. It happened at Nelson Labs. Okay, so this is like, a tale from my past. So what we had a customer. They came to us and they're like, oh yeah, we designed this cauterizing tip for a device that's supposed to cauterize during surgery, right, to limit blood loss. And they were like, okay, now we just need to test it. Okay, so we tested it. We extracted it. Yeah. And because during cauterization, you have that tip being like super hot, right? So we had to kind of mimic that process in that extract. So we did that just to make sure that we are capturing everything that we need in that test sample. So now we did a acute systemic tox test, and what happened? Okay. One of them, so they do sequential injections, so they take the first mouse, and 30 seconds after it was injected, it died. And we were like, whoa, something is going on here, right? So our team, we sat down and we were like, what is happening here? It was like, this is not very common that this happens, right? And because the death came so quickly within seconds, there's only a handful of substances that can do that, okay? And it turns out that this ABS that they were using was leaching cyanide when heated up, because turned out that they are using ABS that is meant for car bumpers, okay? So know your materials, okay? So cautionary tale, just remember that, okay? If tomorrow you'll be like, oh yeah, I remember Helen saying about this ABS thing. Yeah, that's my goal. Just remember it, okay? So when do we need to start thinking about it? There, at the beginning, right? Okay, so when we think about materials, okay, what do we need to know about them? I mean, everybody knows, this is also probably like ABS, right? I mean, it's like Lego pieces are from ABS, right? So you would expect it to be okay. But you need to know that it's a lot of these polymers, they have their backbone, which can be, the same from one manufacturer to another, but there's lots of other stuff in there, right? So what you need to know is you need to look at the TDS, right? What does it tell you about the material? Is it also something that you internally have used as a manufacturer on something else, some very similar device? Or do you have anything, it's always better to use something that is vetted already, right? Because then you have less of a concern in the end. And also, master files are great tools to know what testing has been already done. But I always have to say, when you just see, oh, it has a master file, know what's in the master file, because I have also seen master files that have only cytotoxicity testing in it, okay? That doesn't get you very far, right? So know what's in the master file, but some master files are very extensive, they're very good, and they have lots of valuable information in there, so please check that before you choose a material, okay? Now, let's talk about changes. So changes can happen in many cases, right? I'm sure you're all aware. There's supply chain issues, right? The curse of the COVID, and that alternate sources to cover those supply chain issues, right? You say okay, we have one primary source, but then we want to qualify a secondary source just to make sure that our production is not halted. Or let's say there's an upstream notification oh, sorry, we're discontinuing this blend of polymer. What do you want to do, right? I mean, hopefully you do get those notifications because sometimes I see that it still happens and nobody notifies, which is bad. Okay, so now all of this leads to how does that change impact the biocompatibility of the final finished device, right? We want to make sure that when you have something that is changing, you do look at it. So what do you need to do? How many times have I heard, well, it's silicone, right? But, I wouldn't want to have the silicone that you use to waterproof your bathroom for an implant, right? Basically, some materials may be suitable for some applications, but not for the others, right? And you need to know that. Yeah, understand that even if it says the same name, especially with these softer plastics, it really, unless you can prove it, you cannot take it as a same. Okay? So basically, when you do have to choose a new material, because let's say a supply chain issue, right? You want to make sure that you compare the specs, right? For example, let's say you have surgical tool that has PBAX, right? PBAX 55D, right? I mean, that's a very common material, okay? And now you're saying, oh, this supplier cannot give me anything anymore. Now I have to look for alternatives, right? And, let's say you find another PBAX 55D, right, from a different supplier. And what I say is that you need to kind of make sure that you still look for limited contact devices. If you have, let's say, USB class 6 testing on the material, much better than nothing, right? Especially if you can prove it's the same, toughness, and it's the same specs from one to another, and your manufacturing process is really not changing. That change then should probably be pretty like to like, right? But if we're having something like we're changing from one new sole to a completely different new like rubber type polymer, it can be a completely different story, right? Because we might not know what is in that other polymer, right? Even with the silicones is a great example, because even the cross-linking properties of different cross-linkers, like vulcanized or not, it makes a difference in the extractables profile, right? And because it makes that difference, that extractables profile then could be hazardous to the patient, right? And obviously, we want to make sure that we know that, again, it's not just the backbone. There's tons of other things in there, right? Like this story about like, oh, nobody's going to allow plasticizers anymore, right? DHP, bad. We have to find something else to replace the DHP, right? And now they're like, well, we have a non-DHP in there, right? That doesn't make it better, right? I mean, they might say it, right? Yes, it's not the bad compound, but actually it could be as bad when we don't know anything about it, right? And the colorants, right? I mean, I made it as a joke of like, oh, let's make it pink. But actually, it can be a really big risk assessment that needs to be done if your marketing people say, well, let's make it pink. Yeah, so just tell them to hold their horses before they make the decision, okay? And as I said, if it's not documented, it didn't happen, right? Always, 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 when you have a change, make sure you do a risk assessment, right? Or have us do it if you don't want to do it. And that when there's equivalence, right, we can show that there is very little risk of changing this PBAX 55D to this PBAX 55D, then you might not need to go down the testing route, right? But it has to be documented. You have to demonstrate to the reviewer that you did that mental exercise, right? Okay, so that's pretty much all I wanted to say. I wanted to highlight the couple key points, right? Understand the risks of your device, right? That's the first thing. You cannot do a risk assessment if you don't understand the risks for your device. And then when you do the evaluation for biocompatibility, always, always, always think about it as a plan. You have to have a written document that shows what you were thinking to come up with the approach. And it should be really the first step, that plan, before you start doing testing, okay? And then biocompatibility should be thought of up front, right? when you start choosing your materials, choose them wisely, okay? And everything that has a change that can potentially impact the medical device, especially the patient contacting portions, make sure you do a risk assessment, okay? And the key, really, I know sometimes I have to like toggle the hats of like, safety versus regulatory risk, right? But I'm here really to support the patient's safety. And this is what we really need to be showing with these biological evaluations, that the device in its form that it's intended to be used is not going to do harm to the patient, right? Okay, so that's all from me. Come and visit me at booth 3135. I love talking about biocompatibility, so, come say hi and be like, oh, I thought that your talk was so exciting. And if you want to hear more or read more, there's also an Esoterra Academy that has tons of stuff on biocompatibility, but not just biocompatibility, sterilization, packaging, and everything else. So please go there and sign up and read from our great experts about advice that they would give. And things to think about when you're doing these assessments and documents. All right, that's it. Anybody have any questions? You can always find me at, sorry. No, it went away. 3135. All right, thanks.